Hello everyone to part 2 of our BIS103 lecture 4. In part 1, what we had done was we had to recap a little bit of our aerobic glycolytic pathway, and we had spoken about some variations on this pathway, specifically looking at what happens under anaerobic conditions, looking at fermentation. But what we have done was to look at only glucose as our substrate for the breakdown of sugars. What we want to do in this part 2 of our video is to look at some other sugars. Right? We don't live off glucose alone, at least I hope you don't. So we're also getting through our food a number of other sugars, and I want to talk about some examples here and how we can break these down and feed them into the glycolytic pathway. So what we want to get out of this video is that in the end, we want to be able to explain sort of the common and distinct pathways in the utilization of carbohydrates, limited, of course, to those that we'll discuss here in class. All right, so let's look a little bit beyond glucose, right? We don't live off glucose alone. How about some of the other sugars? So one prominent example that you will have in your food every day is sucrose, right? So that's your table sugar. It's highlighted here. We already spoke about it when we talked about reducing versus non-reducing sugars. And you should be revisiting that. If this doesn't ring a bell, go back to that lecture and sort of familiarize yourself with how we can distinguish reducing and non-reducing sugars. Right? Sucrose is made up of one molecule of glucose and one molecule of fructose, so it's a disaccharide. And both those monosaccharides are combined with each other through this 1,2 glycosidic bond here. I will not ask you how exactly these glycosidic bonds work. We'll talk a bit about how they can be broken down, how they can be formed, but only in general terms at this later stage. Some of your major resources of sucrose are actually sugarcane. This is mostly what we will get in here in the US. A lot of Europe, for example, will rather use sugar beet as a source of sucrose. Sugarcane doesn't really grow all that well in Europe, it being a colder climate. But sucrose generally is coming from a plant resource. Right? So how can we break it down? First step, and we'll look at this later, would be to break this glycosidic bond into monomers, right? Then the glucose part goes through the glycolysis, just as we discussed in the last lecture. But what do we do with fructose, right? So it's highlighted up here, fructose should look fairly familiar. It actually is very similar to glucose. The difference is, right, it's a ketose, ketone function here, whereas glucose is the aldose that has the aldehyde function here. So we're dealing with ketose. Right? And the way we actually get it into our glycolytic pathway is using a kinase reaction. Right? So here you have simply your hexose, your sugar, the kinase reaction, exactly the same way we argued for its importance for the phosphorylation of glucose. We use a kinase here, we use ATP as a phosphoryl group donor. We phosphorylate fructose at carbon 6. What we're generating is fructose 6-phosphate. That should ring a bell because fructose 6-phosphate actually is one of the intermediates in our glycolytic pathway, right? So with this one simple step, we already have entered fructose into our glycolytic pathway. It's coming in form of its phosphorylated derivative fructose 6-phosphate. This is a simple pathway. It's actually occurring in all cells. Interestingly, though, is that we have a secondary and alternative pathway and how we can bring fructose into the glycolytic pathway. That actually is a major one by which we do this, and this pathway is only happening in the liver. Right? So this is liver specific, and it's called the fructose 1-phosphate pathway. Okay? So again, fructose here coming in from our diet. We use the same kind of reaction. We use a kinase, ATP dependent, but rather than at C6, we're actually phosphorylating at C1 here. Okay. So we're generating fructose 1-phosphate. That is not yet a glycolytic intermediate, so we have to work further. The next thing we do is we're actually helping ourselves with an aldolase. So we do a carbon-carbon cleavage. Similar to what we've seen in glycolysis, we do so between C3 and C4. What we're generating is one molecule of DHAP. That should sound familiar. That was one of our products of the priming phase of glycolysis. And we're generating one molecule of glyceraldehyde. That might sound familiar, but we haven't seen it yet. What we had seen was G3P, right? Glyceraldehyde 3 phosphate. This is not phosphorylated yet, right? This is just the aldehyde. So in order to get it to be G3P, one of our glycolytic intermediates, we have to use another kinase to phosphorylate it. We're making G3P, another glycolytic intermediate. Okay. If you think about the bioenergetics of these two reactions, they're actually the same as glycolysis, right? Here with our pathway from fructose to fructose 6 phosphate, we're using one kinase step, just as we did for glucose. We bring it in downstream of this first phosphorylation, so nothing has changed. And the same is true for our alternate pathway here. We have one ATP to make one fructose 1 phosphate. We have one ATP here to make G3P. We're using 2 ATP. We're bringing in the intermediates downstream. So after the 2 phosphorylation reactions of our priming phase of glycolysis, so the net ATP use actually is the same. Just to orient yourself, what have we done right now, right? We wanted to bring in the alternate sugars into the glycolytic pathway. That's, again, here's your glycolysis, right? Your 10 steps. Like I said, fructose, we have one pathway here in all cells that is coming in right here at fructose 6 phosphate. And then our major pathway that is happening in the liver is coming here at this step at the end of the priming phase, bring it in via intermediates as DHAP and G3P. Right? So keep in mind, right, these intermediates of the glycolytic pathway don't only have to come from these individual enzymes that are in the core pathway, right? This enzyme, for example, here cannot only take the substrates from these previous enzymes, it can also take them from other pathways, right? Pathways intersect, they cooperate. So these are the exact same metabolites. So this enzyme can accept the same metabolite regardless of what pathway it comes from for as long as they are co-located and accessible to each other within the cell. Next up, a few more examples. Up here, I included glycerol. I'm cheating a little bit here. Glycerol is actually not a carbohydrate, but it's important in this context, and we'll see it a little bit later. Glycerol is actually a product of lipid metabolism, lipid breakdown. Why is it not a carbohydrate? Right, here's its structure. Remember our definition of carbohydrates. One, they had to be polyhydroxy. And glycerol certainly fits that bill, right? We have 3 hydroxy groups here. So it's a polyhydroxy, but what's missing is a ketone or aldehyde function, right? There should be polyhydroxy with a ketone or aldehyde function. These are only alcohol functions. There's no carbonyl group. Therefore, it's not a carbohydrate. But we're actually helping ourselves with some very similar reactions as we've seen already on how we can bring in glycerol to the glycolytic pathway. Again, we're using a kinase here. We're activating glycerol through glycerol 3 phosphate, so phosphorylating at carbon 3. And the next reaction you also have seen now several times, we're using a dehydrogenase. We're using our oxidized NAD cofactor to oxidize the alcohol function here at C2. What we're generating is DHAP, our glycolytic intermediate, right? Task achieved, we have brought in glycerol successfully, converting it to a glycolytic intermediate. Another example here, mannose, that is a sugar, it's a hexose, it's an aldose, right? What it actually is a C2 epimer of glucose. So if you want to remember those structures, right? If you remember what glucose looks like, it's your aldehyde, then our hydroxy groups, right, left, right, right. And now here at C1, C2, right here, we have the hydroxy function on the other side. So it's a C2 epimer of glucose, right? Mannose actually is a common sugar, again, in fruit. So you will get it through your diet as long as you eat your fruits. What do we do with it? Same procedure here. is that we are bringing in a kinase, we're phosphorylating at C6, making mannose-6-phosphate, same kind of activation as we have seen for fructose and for glucose. And then in order to make this a glycolytic intermediate, we're using an isomerase, very much similar to the isomerase we used in reaction 2 of glycolysis. We are converting the carbonyl group here at C1, we're moving it to C2. What are we generating here is fructose 6-phosphate, our glycolytic intermediate. So job done, we have brought in mannose into the glycolytic pathway via fructose 6-phosphate. Again, for your orientation here, mannose right up here, coming into glycolysis via fructose 6-phosphate, and glycerol, remember, it's not a sugar, but it's important in this context to try and understand where it's coming in. It's coming in here at dehydroxy adenine phosphate, right? All right. One more example here, another sugar that you will encounter, especially if you enjoy sort of your daily dairy, milk, cheese, yogurt, and so forth. They contain lactose. It's another disaccharide. Right? In this case, it's a disaccharide of a monomer of galactose here and a monomer of glucose. Right? Sort of as a fun fact at the side, those of you who suffer a bit from lactose intolerance, what is actually happening here is that lactose, if you take it up as a dietary sugar, usually you would have an enzyme, it's called lactase, that is capable of breaking this bond in the disaccharide and to release the free glucose and free galactose. Right? Not everybody has that. It is actually more common in cultures that more heavily rely on dairy in their diet. So if you have a reduced activity or reduced amount of this enzyme, of the lactase, you cannot break this glycosidic bond. And what will be happening is that then actually the bacteria in your gut will start to ferment this sugar. And this causes all the sort of the issues in your gut and the gases that come with lactose intolerance. Right? So how, assuming we can break the glycosidic bond, how do we bring this in? It's actually a tiny bit more complicated than the others, but I will go through it just as a general mechanism, and I will only ask you to really know this general understanding, not the mechanistic details of it. So here you have your galactose. We have broken the bond. Glucose has gone into glycolysis, just as we had discussed. What do we do with galactose? First off, what is galactose? This actually is now the C4 epimer. So C1, 2, 3, 4. The hydroxy group is facing in the opposite direction as we know it from glucose. So it's a C4 epimer of glucose, just to make it a bit simpler for you to remember these structures. Shown here now in the Haworth production, that's again galactose. First step looks familiar, right? We're using an ATP-dependent kinase, and we're phosphorylating galactose at C1 right here. So far, so good. That seems now familiar from all the past examples I showed you. But now we're not actually using other steps like an oxidation reduction or an isomerization. We're actually helping ourselves using a transferase type reaction. And so what we're actually bringing in is a molecule of UDP glucose right here. That's basically a co-substrate in this reaction. We get this molecule from nucleotide metabolism. We'll not touch much on it this quarter because it's just getting too far ahead of ourselves. So for now, just assume it's coming from nucleotide metabolism. That makes sense if you look at its structure here, right? You have a base here, you have a ribose sugar, you have a phosphoanhydride, and then a sugar. So this looks a whole lot like ATP in its structure. So it's very similar, but instead of carrying a third phosphate group here, or a niacin group like we'd seen in NAD, it's now actually carrying a molecule of glucose. And so just to sort of highlight what is happening here, I have glucose circled in red and your galactose circled in blue. What this transferase is doing now, it's simply switching glucose for galactose on this nucleotide type backbone, okay? So this is the result. We're switching over, we're bringing in galactose, we're attaching it here this bond is broken, glucose is released as phosphate. We are cleaving here at the high energy releasing phosphate group. We're bringing in galactose. So now we have UDP galactose, and our product is glucose. Okay. In a way, it's equivalent to an isomerization, but it happens to be that for galactose, it's really difficult to achieve this isomerization. We simply don't have that enzyme. So we go around that problem by using a transfer, bringing in glucose and transferring it over. We're releasing glucose 1-phosphate. And this now we can use an isomerase for, and we can generate glucose 6-phosphate. Right? So galactose actually coming in at a different step and via a slightly different mechanism. Right? So galactose here for your orientation, coming in at the second step here at glucose 6-phosphate. Okay. So this is a chapter on alternate sugars outside of glucose. We will continue this discussion both for the utilization and the storage of sugars in our next lecture, in lecture 5, when we start to talk about polysaccharides.